you guys
are
recording.
So,
Okay.
we start off with glucose. Cellular respiration starts off with glucose, C6H12O6. In the cytosol, it goes through glycolysis. Comes out, we get 2 pyruvic acids and 2 ATP as well as 2 NADHs. Now, then you get to this big question, is there oxygen or not? If there's not oxygen, we're going to go to anaerobic respiration, which also all takes place in the cytosol. There are 2 different types of anaerobic respiration. Alcoholic fermentation, which will convert the 2 pyruvic acids into ethyl alcohol and carbon dioxide. As a result of converting the pyruvic acids into ethyl alcohol and carbon dioxide, the 2 NADHs that were made in glycolysis will be converted back to NAD+. That NAD+ can be used in glycolysis to help convert the glucose back into pyruvic acid, or into pyruvic acid. There's also lactic acid fermentation, where the 2 pyruvic acids get converted into lactic acid. In doing so, the 2 NADHs that were made in glycolysis again will be converted into NAD+, which will allow glycolysis to keep running, keeping the cell alive while there's no oxygen available. Kind of summed up right here. The 2 ATPs will help keep the cell alive, so it'll use these 2 ATPs to help keep the cell alive. Whatever, any little thing that will keep the cell alive that needs energy, it'll go to that. And like I just said, the fermentation changes the NADHs made in glycolysis back into NADH, keep the glycolysis running. Okay, if there is oxygen available, the 2 pyruvic acids will get converted to acetyl-CoA. And that takes place in the mitochondrial matrix. The mitochondrial matrix is the innermost part of the mitochondria. It's inside the inner membrane. Now, by converting pyruvic acid into acetyl-CoA, which is done by coenzyme A, you also get you also make 2 NADH molecules. So far, we've actually produced 2 NADH molecules from glycolysis and 2 NADH molecules from converting the pyruvic acid into acetyl-CoA. So far, we already have 4 NADH molecules. We'll get back to that later. Acetyl-CoA, once it's made, it combines with oxaloacetic acid, which is spelled wrong, into the citric acid. So you have 2 carbon compounds, 4 carbon compound becomes a 6 carbon compound. Then it goes through the pathway. We're not going to go over it in great detail, it's not really necessary. But within 2 turns of the Krebs cycle, for each one acetyl-CoA, you get 2 FADH2 molecules, 6 NADH molecules. You get some carbon dioxide, which just diffuses out of the cell and into the blood, so it can be exhaled. And you also get 2 ATP molecules. Once the Krebs cycle is done, we take the 4 original NADHs that we had from glycolysis and converting the pyruvic acid into the acetyl-CoA, plus the 6 that were made in the Krebs cycle. So we have 10 NADH, and we also have the 2 FADH that are made in the Krebs cycle. They're both, well, all of them are going to the inner mitochondrial membrane, where they're going to go through the ETC, the electron transport chain, and chemoosmosis, hemiosmosis. So they do their thing there. They're giving up their protons. So the ATP synthase can do its thing, and eventually, after all is said and done, each one of the NADHs that we're breaking down will give us 3 ATP. Each one of the FADH2s will give us 2 ATP. So with the 10 NADHs that we had, 2 FADH2s, and the 4 ATPs, we get a total of 38 ATP produced from the complete breakdown of 1 glucose molecule when oxygen is available. You may also be wondering what happens to the electrons and the protons at the end of the electron transport chain. Because as we know, electrons can't get onto the electron transport chain until the ones that were before them get off. So reason why we breathe oxygen gas is it acts as the final electron acceptor to these electrons and protons. They all combine, you get one oxygen molecule, one molecule of O2 plus 4 electrons plus 4 protons gives us 2 molecules of H2O.